Hi everyone, Dr. Ahles here. In this short video, we're going to talk about the kind of macromolecule called carbohydrates. When we talk about carbohydrates, big picture, their primary function is to provide energy for our cells. Many of them either are glucose or can be broken into glucose, which is our cell's favorite kind of food. It's the easiest kind of energy. We find carbohydrates in all of the things that they tell you to cut when you're doing a low-carb diet. So it's found in grains, we find them in fruits and vegetables, naturally occurring, but big picture, their job is to end up as glucose in our body. When we talk about carbohydrates, there are three groups that they come in. The most simple kind of carbohydrates are called monosaccharides. Mono means one, so the monosaccharides are the smallest pieces that we find of carbohydrates. If I attach two monosaccharides together, I end up with what's called a disaccharide. A disaccharide is made out of two sugars instead of the one sugar of monosaccharides. The final kind of carbohydrate that we have in the body are called polysaccharides. And when we talk about polysaccharides, we've put together many of the monomers. Many monosaccharides, many disaccharides. Polysaccharides are the biggest type of carbohydrate. When we talk about monosaccharides, the single sugars, there are several different varieties that we see. But for the most part, monosaccharides are going to have 3 to 7 carbons in their backbone. And you can see over here on the side where we have carbons in each of these monosaccharides. Now, monosaccharide names are always going to end with "-ose", and sometimes the beginning of their name would tell us how many carbons they have. So for example, we have some sugars in the body that are trioses, meaning that they have 3 carbons. Other sugars in the body are pentoses, and a pentose is something that has 5 carbons. So by that pattern, a hexose, like a hexagon, has 6 carbons. When we look at our monosaccharides, which again are the pieces we'll use to build disaccharides and polysaccharides, we see 2 of those functional groups that you're learning. The first functional group is a carbonyl group. So the carbonyl groups are a C with a double bond to an oxygen. You can see in our monosaccharide image over here, each of my monosaccharides has a carbonyl group. We also see in our monosaccharides many different hydroxyl groups. Remember that a hydroxyl group is an OH group. So in each of these types of sugars, whether you have 3 carbons, 5 carbons, 6 carbons, we're always going to have carbonyls and hydroxyl groups. Up to this point, we've looked at monosaccharides in what we call their linear form, but especially if we're talking about a monosaccharide that has 5 or 6 carbons inside of it, a lot of the time you're actually going to find that in the ring form. So as we start looking at specific monosaccharides, you'll notice that many of those will be presented in the ring form. So my first example of a monosaccharide that typically exists in the ring form is called fructose. So fructose, as in high fructose corn syrup, that molecule, fructose is an energy source that is made by plants. It has 5 carbons in it instead of 6 carbons in it, like our typical energy source. Another 5 carbon monosaccharide is ribose. Ribose sugar is what I use to build ribonucleic acid, also known as RNA. Ribose sugar has 5 carbons. Deoxyribose, which is in DNA, is actually the same as ribose sugar. We just take away an oxygen. So deoxyribose and ribose sugar in your nucleic acids, these are our 5 carbon monosaccharides. We can't forget arguably the most important monosaccharide, and that is glucose. Glucose, again, is your cell's favorite food. So it's a 6 carbon structure, usually exists as a ring, and glucose is what we use to make ATP, to make the energy power for our cell. When I combine together some of my monosaccharides, I can build disaccharides. Disaccharides are connected to one another using covalent bonds, and I actually have a special name for the type of covalent bond, the type of covalent bond that I make in disaccharides. That kind of bond is called a glycosidic bond. And once you write down that word, I want you to underline, highlight, star it. Glycosidic bonds are only seen in carbohydrates. And a glycosidic bond is made through a dehydration reaction. Remember that in a dehydration reaction, I steal things from the two monomers I'm connecting. Those things come together to build me a molecule of water and also to build me a covalent bond. So a glycosidic bond is the type of covalent bond that forms between the two monomers of a disaccharide. When we look at disaccharides, there are three specific examples I want you to be familiar with. The first kind is called sucrose. Sucrose is a disaccharide when we group together glucose and fructose. Both of these come from plants, glucose and fructose. And actually, when we talk about sucrose, this is the chemical formula for the sugar that you have in your pantry that you use to cook with. So sucrose is a polymer. It's made of multiple pieces. Those two pieces are glucose and fructose. Notice, by the way, you see the note here that says it's a glycosidic linkage or a glycosidic bond. Two sugars being connected to each other. The next disaccharide for you to be familiar with is maltose. Maltose is what forms when I have two glucoses that are attached to each other. Again, we're using that glycosidic bond to attach our glucoses together. Maltose is usually a product of when we're breaking down starch. Starch is lots and lots and lots of glucoses connected to each other. When I chop them into little pieces that are too big, I've generated what's called maltose. And then I'll use an enzyme to go through and chop the maltose into little pieces as well, into glucose pieces. One final disaccharide that you'll definitely have heard of before is lactose. Lactose is the kind of sugar that's naturally found in milk. If someone doesn't have the ability to chop up lactose into glucose and galactose, we would say that they are lactose intolerant. This particular sugar, they don't have the ability to chop up that glycosidic bond. So lactose, one other example of a disaccharide, meaning it has two pieces being used to build it. We also in the body, though, have many, many things that are bigger than just two pieces. When I'm talking about things that are bigger than two pieces, we call them polysaccharides. Many pieces put together, many sugars. Polysaccharides are a good way for us to store energy. That stored energy can be chopped up and we can get to it relatively quickly compared to how long it would take me to get to lipids, which we'll talk about next. When we're talking about polysaccharides, in addition to being a good energy storage, they're also very good for giving me the structure or the shape of things, for example, the body of insects. So multiple different functions that these large sugar compounds perform. Let's talk about the first kind of polysaccharide that we're discussing, and that is starch. Starch is a polysaccharide made out of lots and lots of glucose. So when plants build glucose, they then transform that glucose into starch. We can either have glucose molecules that exist in a straight line, which is really easy for us to chop up right away, or we can put them into a branching pattern, which we can also chop up. So starch, made of many glucose monomers, existing in a linear form or in a branched form, our first polysaccharide. Our next polysaccharide is also made by plants. This polysaccharide is called cellulose. Cellulose is something that we find in the cell wall of plants, giving them their boxy shape, if you will. Cellulose has glucose molecules that are alternating their direction. So each of my glucose molecules has a little hydroxyl that sticks out from its rings. If I alternate which side of the structure I have that hydroxyl group, that gives me something that's very stable and can exist in a straight line. If they're right next to each other, I can't get this straight line arrangement like I usually have. The thing with cellulose is that most animals, including humans, don't actually have the enzyme that's used to break down cellulose. So we have the specific enzyme called cellulase that would chop up cellulose for us, except we don't have it, most animals don't have it, but what do have it are animals called ruminants. And ruminants are animals like a cow or other grazers like deer. The only reason we would say that they have cellulase is because inside their digestive tract, they have bacteria that have cellulase. So as they're chewing on the grass, as they're chewing on plants, those bacteria are able to access that cellulose and chop it up into glucose that those animals can then use. But for the most part, humans are not able to digest this polysaccharide called cellulose. Another example of a polysaccharide is chitin. Chitin is a polysaccharide occurring naturally in both bugs, making up their exoskeleton. So if you've ever seen those little leftover body shapes of cicadas when they're molting, we also see chitin inside fungi, which are things like mushrooms or the kind of fungi that can give you a fungal infection. We as humans don't naturally have chitin in our body, so we can actually use chitin as a target for antifungal medication. It won't affect your cells, it'll only affect the type of cells that have it, which are the fungus that's giving you an infection. Chitin is very similar to cellulose, so it'd be another thing that humans couldn't break down. But when we talk about chitin, we've added an extra nitrogen. So we've got some amine groups that are showing up here inside chitin. That's the only thing that makes it different from cellulose. So we still have our functional groups on opposite sides, and we're adding in that nitrogen to give it a different kind of texture. The final polysaccharide that I want to mention for you is called glycogen. And glycogen is probably the most common polysaccharide when we're thinking about who uses it. So animals like humans, as well as the fungi, as well as bacteria, all use glycogen as a way to store their glucose. So we store glycogen in our liver and in our muscle tissue. And the function of it in those places is to provide a relatively quickly accessible stored energy. So I told you we store it in our liver and we store it inside our muscles. What happens is when your blood sugar gets low, we send out a message to the liver, or when we're exercising, we send out a message to the muscle saying, hey, you've got some stored glycogen. I need some glucose. Chop it up for me and give me that glucose. So the liver can be used to release glucose into the bloodstream, whereas in a muscle cell, if I chop up my glycogen, that will give me the energy I need to do a muscle contraction right away. So of all the polysaccharides, the one that exists inside humans is glycogen, allowing us to store some energy. The other polysaccharides that we talked about, like chitin or cellulose, we see those in plants, we see those in fungus. Those help them primarily to do things like bring structure to themselves. On that note, we're going to wrap up our discussion of carbohydrates. Next up for you, the video about lipids, the other way that our body can store energy.